In this video lesson, we will learn about non-Mendelian inheritance, and the learning goals to be achieved are: 1. Explain how the genotypes of an incomplete dominant trait affect the phenotypes of the heterozygous offspring. 2. Explain how the genotypes of a codominant trait affect the phenotypes of the heterozygous offspring. 3. Distinguish between complete dominance and codominance. 4. Describe complex patterns of inheritance, including multiple alleles, Pliotropy, and polygenic inheritance. And 5. Use knowledge of the human ABO blood group system to solve paternity problems. In my previous two lessons, we explored the Mendelian principles of inheritance with his three laws: law of dominance, law of segregation, and law of independent assortment in pea plants. All of Mendel's work were done on traits that could independently segregate, assort, and express dominance and recessiveness. Mendel's fundamental work on pea plants opened the doors to other scientists to explore genetics. As more of the understanding was made by other scientists on different plants, they discovered that not all traits or genes behave in a way that can be explained with simple Mendelian inheritance. Today we know that the inheritance of many traits or genes are extremely complex. So in this video lesson, we are going to learn about some of the traits that show non-Mendelian inheritance. So let me first explain to you inheritance of flower color in Snapdragon plants. These plants are scientifically called as anti-rhinum. So scientists conducted similar Mendelian monohybrid crosses in Snapdragon. They took true breeding red flowered plants and crossed them with true breeding white flowered plant. The genotype that I'm giving for these two parents are: red flower, we have 2 capital R homozygous, and for the white flower I'm taking 2 capital R with dash, R dash homozygous. Okay, now when such true breeding plants were crossed and they obtained the first filial generation, the genotype of all of the F1 generation had a heterozygous RR- combination. And when they checked into the phenotype of the flower color, what they saw was all of the F1 plants had pink flower color. Now this was a completely different result from that of Mendelian monohybrid result. So here F1 plants did not had the flower color of either of the parents. It was neither red nor white, but they seem to be showing a kind of a blended color in the F1 plants with pink coloration. So now the question was, is it really a blending inheritance that we can be explaining for Snapdragon flower color inheritance? To get more of the confirmation on this pattern of inheritance, they went forward with F1 selfing in order to study the second filial generation or the F2 generation. So on selfing, they obtained F2 generation that had 3 different types of genotypes. So the 3 different genotypes were RR homozygous, RR- heterozygous, and R' homozygous. And this occurred in a ratio which was 1:2:1. So out of the total of the F2 population, 1/4 of the population had RR homozygous genotype, 2/4 of the population had RR- type of heterozygous genotype, and 1/4 of the population had R' homozygous genotype. And this you can be easily predicting or even finding here in the Punnett Square. Now, when the F2 phenotypes were observed, what they could be surprisingly finding here was, apart from the reappearance of pink color, they also had pure red and pure white colored flower in the F2 generation, very similar to that of the red color and the white color in parental generation. So the F2 generation of the total population, 1/4 of the population had red flower color, 2/4 of the population or half of the population had pink flower color, and another 1/4 of the population had white flower color. And the phenotypic ratio was also 1:2:1. So you can find in the inheritance of Snapdragon flower color that the genotypic and phenotypic ratio is the same. genotypic ratio is also 1:2:1 and phenotypic ratio is 1:2:1 as well. Basing on this result, the notion that appearance of a pink color in the F1 hybrids could be a blending inheritance was completely disproved because of the reappearance of pure red and pure white parental colors in F2 generation. So now what could be the inheritance here? This pattern of inheritance is completely deviating from Mendelian inheritance because in the heterozygous combination, capital R, which is the allele of red, is not completely dominant over R dash, which is the allele for white flower color. So red color is not completely dominant over white color, nor white color is completely dominant over red color. And because of which we are not able to be seeing one of the dominant traits of the parent in F1. So what was explained here was that both of these alleles, the allele for red color and the allele for white color, R and R dash, are incompletely dominant over each other. So red color allele R, capital R, is incompletely dominant over R dash. Or we can also say R dash is incompletely dominant over capital R, and because of which in the heterozygous combination, because of the partial expression of the allele. Okay, so we have a partial expression of the red allele and the white allele was not completely recessive. So we also have a partial expression of the white allele, and that kind of the incomplete partial expression of the alleles resulted in an intermediate phenotype, a pink phenotype, which was a new phenotype not seen in both of the parents. And this kind of the inheritance is referred to as incomplete dominance. Incomplete dominance. So this is how we have the explanation to the inheritance of flower color in Snapdragon, where it doesn't follow Mendelian law of dominance, nor it is a blending inheritance. But here the inheritance of flower color is said to be an incomplete dominance. An incomplete dominance pattern of inheritance where both of the alleles are partially or incompletely dominant over each other, and as a result of which we have the heterozygote expressing an intermediate or a different phenotype to that of the parental phenotype. So here is another example of the inheritance of feather color in chicken. Now you can be seeing a similar pattern as it was or in the case of flower color in Snapdragon. So when a true breeding black feathered chicken with BB, capital BB genotype was crossed with a true breeding white feathered chicken with the genotype capital W, capital W, what they observed in all of the F1 generation was that all of the F1 had a heterozygous capital B, capital W genotype, and all of these F1 chicken had blue feathers. So it is again a very different phenotype or a feather color as opposed to the black and white feather color in the parents. So they had a new phenotype which was blue feather color. And when such F1 blue feathered chicken were crossed with each other, then in the F2 generation, the genotype were again of three different types: homozygous capital B, heterozygous capital B, capital W, and homozygous capital W in the ratio 1:2:1. And the phenotypes of such F2 chicken had black, blue and white phenotype or white feather color in the ratio 1:2:1. So again you can be seeing the genotypic and phenotypic ratio of the F2 generation remaining same here with 1:2:1 ratio for genotype as well as 1:2:1 ratio for phenotype. So this is again an example of an incomplete dominance where capital B is not completely dominant over a white, nor white is completely recessive or completely dominant over black. And as a result of which we have the expression of an intermediate phenotype in the heterozygotes. Okay, so the law pertaining with the inheritance of flower color in Snapdragon or in other flowers as well, like in 4 o'clock plants, as well as the flower color in carnations, all exhibit this pattern of inheritance, which is known as incomplete dominance. So incomplete dominance can be defined as a form of intermediate inheritance in which one allele in a heterozygote, one allele in a heterozygote is not expressed completely over its paired allele. And such kind of the inheritance pattern is incomplete dominance. To explain another deviation from Mendelian inheritance, let me take the example of the inheritance of AB blood group in humans. So in the human population, we know that there are 4 different blood types: AB and O blood type. In the inheritance of AB blood type, we see a kind of a deviation from Mendelian inheritance. Let's assume that we have 2 parents who are homozygous for blood type A and blood type B. Now, blood type or blood group gene is denoted with capital I letter, and the alleles for the blood type gene is shown as a superscript. So, a parent with homozygous A blood group has a genotype which is capital IA combination, and another parent with the homozygous B blood group has capital IB homozygous genotype. Now, from such parents, when we look into the possible genotypes and the phenotypes in the progeny, so what we find is the possible genotype in all of the progeny is going to be a heterozygous IA-IB combination. And such kind of progeny with the heterozygous genotype are to be expressing only one phenotype, but this phenotype is again very different from either of the parental phenotypes. It is neither A nor B a blood type. So these F1 progeny have the phenotype which is of AB blood type. So you can see a difference from the Mendelian monohybrid result. And this kind of the inheritance of AB blood group from the parent to the progeny in the human is another form of deviation of Mendelian inheritance. Now, how do we explain with this pattern of AB blood group inheritance? What we can see here is in the heterozygote, when the allele IA and IB are both existing together, in this state, when we look into the phenotype has the expression of both of the alleles trait. It is AB. So, which means to say that allele IA can also express in the presence of allele IB. And in the same way, allele IB can also make its expression irrespective of the presence of the other IA allele. And because of which, this kind of the inheritance is referred to as codominance. So this pattern of inheritance in AB blood type is a codominant inheritance pattern. Now, what do we mean by codominance inheritance? Where we have the inheritance in such a way that both of the alleles in a gene, are able to be simultaneously expressing, are able to be completely expressing when they are present in a heterozygote. Okay, and this phenomenon is codominance. So it is a form of inheritance in which both alleles of a gene. Now here in the example, the gene is I gene, that is blood group gene. So both alleles of a gene can simultaneously express. So they express together. Okay, so co means to exist together. So we have simultaneous, can simultaneously express in a heterozygote. So this is how we can be explaining with the nature of inheritance in AB blood group that is there seen in humans. Here is another example of the cross between a red and white coat colored cow. So when a red coded and a white coded cow are crossed together in the F1 generation, all of these hybrids are going to be having a heterozygous genotype of the combination capital R, capital W, heterozygous genotype, and the phenotype of these cow are going to be roan. Roan is a different phenotype to that of a pure red and a pure white parental coloration. So roan is a mixture of red and white. So you can have a kind of a patchy appearance in the F1 hybrids. And when such F1 hybrids are served in the F2 generation, we are able to find three different types of genotypes in the ratio 1:2:1, where the three genotypes are homozygous RR, heterozygous RW, and homozygous WW. And the phenotypes of, or the coat color in the F2 generation are going to be red, roan, and white. And this appears again in the ratio 1:2:1, where 1/4 of the cow are going to be having the pure red coloration. Then 2/4 or half of the cow population are going to be having roan coloration. That's a kind of the patchy coloration, and then 1/4 of the cow can be having pure white coloration as it was in the parents. So 1:2:1 is the F2 phenotypic ratio, and 1:2:1 is also the F2 genotypic ratio. But the difference here is in the heterozygotes, we can see the expression of both of the alleles, complete expression of both of the alleles. That is to say, you can see in the roan white as well as red patches. So white is able to make its expression in the presence of red allele. In the same way, red is able to make its expression in the presence of white allele. And hence you have the phenotype, which is a new phenotype, but then it is a phenotype which has both of the phenotypes of the parental cow. And such kind of the inheritance is known as codominant or codominance, where capital R or the red is able to be making its complete expression in the presence of capital W, which is white. So both of these are dominant and both of these are equally able to be expressing themselves in the presence of each other. So we have a kind of the expression of both of the alleles, and this is called as codominant. The third deviation that I would like to discuss here is multiple allelism. So as Mendel proposed, each of the trait is controlled by a pair of factors. That is to say, each gene is controlled by a pair of alleles. But now what we understand is, in an organism, we have many genes that are controlled by more than a pair of alleles. And one such example is the human ABO blood group system. So our ABO blood group system is controlled by a gene which is known as capital I gene, and this gene is located on our chromosome number 9. Now this capital I gene exists in three different forms. So the three alleles of I gene are capital I, capital A superscript, capital I, capital B superscript, and a small I, or sometimes this is even known as O allele. So these are the three alleles of I gene. And this phenomenon where we have the state of having three or more than three alleles for a single gene is referred to as multiple allelism. And these alleles are referred to as multiple alleles. Now the reason why we have three alleles existing in the I gene is depending on the presence or absence of two of the proteins or antigens, which are known as A antigen, capital A antigen, and capital B antigen. Or these are the two different types of glycoproteins which are present on the surface of RBC. We have already learned these things in our previous class 11 chapter. So now, since we have three alleles, okay, so allele capital I, capital A superscript has the A antigen because it has A glycoprotein on the surface of the RBC. It's called as capital I with capital A superscript. Then IB alleles has B antigen. And because of which it is given the name capital I, capital B superscript. And small I allele or the O allele does not have any of these antigens. So there is no antigen on, they don't have the A antigen nor the B antigen on the RBC surface. Now, by looking into such possibility of the 3 alleles in a population, we know that an individual will always be receiving a pair of allele, right? One each from the parents. So if we try to be looking into the possible genotypes by combining, having a kind of a random combination of the 3 different alleles from each of the parents, we get these possible genotypes. Okay, so this is a Punnett Square where we work out with all the possible genotypes. Now, if you look into the 9 boxes here, you can be observing that in all of these possible genotype, we can have 6 major types of genotypes. And these 6 types of different types of genotypes can be then expressing in 4 different phenotypes in human population. Okay, so in a human population, we can be expecting humans with A blood group, B blood group, AB blood group or O blood group. And such phenotypic expression of ABO system depends on the genotypic combination in the individual. Okay, so now let's look into the possible genotypes for A individuals. So individuals with A blood group are going to be having they are to be expecting to be having the IA allele. Okay, and if you look into the genotype here, we can find that IA allele can be existing in homozygous combination, or it can also be existing in heterozygous combination. And whether it exists in homozygous combination or in a heterozygous combination, since IA allele is a dominant allele, it is completely dominant. Okay, so IA allele and IB allele, because they are written in capital letters, so we need to know that both of these alleles are completely dominant over the third allele, which is small I allele. Okay, and in a heterozygous IA small I combination, we can be expecting the expression of A phenotype. So we can say that for A individuals, there are 2 possible genotypes, which can be a homozygous IA genotype or a heterozygous IA small I genotype. Likewise, for B blood group individuals, again they can be having 2 types of genotypes. The 2 types of genotypes are homozygous IB type of genotype and heterozygous IB small I genotype. So 2 possible combinations, homozygous and heterozygous IB small I genotype. Now if we look into the genotype of AB individual, since we have learned in the previous understanding of the inheritance of codominance, where we said that both the alleles, allele A and allele B, are equally dominant over each other. So IA, IB have to be in heterozygous condition, and in the heterozygous condition, both of these can be making their equal expression. So we have AB individuals always to be having only one genotype, which is to be heterozygous IA, IB. And for O individual, the only genotype that they can be having is small I genotype, because as I said, this third allele small I is written in the lowercase letter because it is completely recessive. It is completely recessive to IA, it is completely recessive to IB, and because of which, if it needs to be making its expression, they are to be existing in homozygous state. So O individuals are always to be expecting a homozygous recessive genotype. So in this way, the 6 different types of genotypes can be explained into 4 types of, or can be denoted by 4 types of possible phenotypes. A with 2 possible genotypes, B with 2 possible genotypes, AB with only 1 possible genotype, and O type with only 1 type of genotype. Well, let's now study the pattern of inheritance of AB and O blood group in human population. So from our understanding about the genotype and phenotype, we realize that A individuals can be having two possible genotypes, a homozygous A genotype and a heterozygous IA small I genotype. Now from this heterozygous combination and the expression of only one phenotype of A blood type, we now know that IA allele is completely dominant over small I allele and small I allele is completely recessive over IA allele. And because of which we have the expression of only the dominant allele. And hence this A blood group in human follows typical Mendelian inheritance. Likewise, if we look into individuals with B blood group, now again these individuals can be having two possible genotypes. It can be a homozygous IB genotype or a heterozygous IB small I genotype. And since a heterozygous IB small I genotype can be expressing only B phenotype, again we see a complete dominance and a complete recessiveness kind of inheritance, and hence blood group B inheritance is even a Mendelian inheritance. But when we think about AB blood type inheritance, now in this or in these individuals with heterozygous, they only have a possible genotype which is heterozygous IB-IA genotype. Now in this only IB-IA genotype, we can find that both of the allele IA as well as IB are able to be making their expression in the phenotype. Which means to say that these individuals are able to be producing the glycoproteins or the antigens, two different antigens on their RBC surfaces, and because of which they are AB. And this phenomenon where both of the alleles are equally dominant and are able to be completely expressing themselves in the presence of the other allele is what we discussed earlier as codominance. So here the inheritance is deviating from or different from Mendelian inheritance. AB individuals inherit the trait through a codominant inheritance pattern. And the last one with O blood type individual. Now these individuals are to be having only one genotype, which is homozygous recessive. And again, we know from Mendelian concept that if a recessive allele is to be making its expression, it needs to be always in a homozygous combination. And because small I exists in a homozygous combination, and because it is in the absence of the dominant allele, it's able to be making its expression, that is the O blood type. And so we realize that O blood group inheritance is again the Mendelian pattern of inheritance. So from this we can conclude that out of the four blood type in human population, blood type A, B and O follow with Mendelian pattern of inheritance, which is to say they follow the law of complete dominance and law of complete recessiveness, whereas AB blood group individuals or AB blood group inheritance follow another type of inheritance known as codominance. Now you might ask why we need to know our blood group. This is vital because at the time of blood transfusion, we need to make sure that we always receive a compatible blood type. Now that is because our immune system is very efficient in terms of differentiating between self-antigen and foreign antigen. So our immune system will never be producing antibodies against our own self-antigen. But whenever our body encounters a foreign antigen, our immune system is immediately going to be producing lots of antibodies in order to counterattack the foreign antigen. So when there is a chance of mismatched blood transfusion, at that time it can be resulting in antigen-antibody reaction, or it can be resulting in immune reaction or immune rejection. Okay, which can be even causing with the clumping or the agglutination of the blood, and sometimes it can be even resulting into fatal consequences. Now in this context, I want to be having a brief the explanation on two major types of the blood type here. AB blood type is also referred to as universal recipient, and O blood type is referred to as universal donor. Now we have learned already about these in our previous class. So universal recipient. This is because since AB has both of the antigen, they have A antigen as well as B antigen, so it doesn't make much of the difference in terms of receiving blood from any of the different blood type. O individuals are, O blood type is a universal donor because since they lack any of the antigen, it will never be causing any kind of an immune reaction. Okay, now you might also ask me, what about A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative type of blood reference. So now this refers to another system. The positive and negative factor that we write over the letter, it refers to another system of blood grouping, and that system is known as RH system. Now RH is a kind of a protein which is also present on the surface of our RBC. And this protein, which is the RH antigen, if it is present on the surface of our RBC, then we become A positive. Let us say I have A antigen plus I also have RH antigen. So I become A positive. In another way, if I have B antigen and I also have RH antigen, then I become B positive. Likewise, if I have both A and B antigen and the RH antigen, then I become AB positive. Likewise, if I don't have any of these two antigen, but if I have RH antigen, then I become O positive. And in that way, if I don't have it, I become the RH negative for all of these 4 different types of blood. So in this way, we can be expecting, basing on these 2 systems of blood grouping, we can be expecting 8 different types of blood groups in human population, which are A positive, B positive, AB positive, O positive. Likewise, we can have the negatives, A negative, B negative, AB negative, and O negative. So in that way, we have 8 total different types of blood groups in human population basing on the 2 system, that is ABO system. So all these letters are denoting with AB antigen, presence or the absence of the AB antigen, and the positive and the negative sign denotes with the presence or the absence of RH antigen. Okay, so now whenever we need to be making a blood transfusion, it has to be ensured that it is always a compatible donor. Okay, a recipient can never be, for example, an A positive recipient cannot be receiving blood from any of the donor. It has to be a compatible donor's blood, which can be A positive, it can be A negative, or it can be O positive or O negative. Likewise, we can be also thinking for all the other types of blood and their respective compatible donors. Okay, and that's the reason why it's very important for us to be knowing with our blood type, so that during the emergency situation, we are able to be having the correct compatible blood transfusion. With the knowledge on the human blood type, we can now be solving any of the blood type genetic problems. Okay, so what I'm going to be doing is I'm going to be working out one such problem for you to give you the idea of how it has to be done. Now, I have a question here. 2 people, both with AB blood, have 4 children. Okay, so this is the situation that is given. 2 people, both with AB blood, have 4 children. What blood types should the children be? So we are expected to be knowing the phenotypes of these 4 children. Okay, so to work out, let's start with the parent. Now, in the given question, we have both of the parents having AB parent. Okay, and now we need to write the genotype. And for AB, there is no confusion because AB individuals have only one genotype. So it's going to be heterozygous IA, IB for this AB parent, as well as IA, IB for this parent. Then we can show with, so this is parental generation. Now you can show the gametes. So 2 different gametes from each parent. And now we can predict where the genotypes and the phenotypes of the next generation by using Punnett Square. So let's use the Punnett Square. Let's call this F1 generation, and I'm going to be drawing Punnett Square. And since there are 2 gametes, you know I will be having 4 boxes. I will write the gametes, parental gametes, one on top, one on the side. Then I will make all the possible genotype combination as per the parental gametes. And here I'm going to be making it more consistent. So I have IA always first. So I'm going to be writing IA first and then followed by IB. Okay, just to make it more consistent. And then I have IB, IB. So these are very easy for you to do because you have already worked out with Mendelian crosses. Now, let's look into the Punnett Square. So there are 4 possible genotypes for the F1 or for their children. So what are the possible genotypes? Let's note down the F1 genotypes, where we have homozygous IAIA, heterozygous IAIB. 2 of the boxes have the same heterozygous genotype, and one more box have homozygous B genotype. So we have 3 types: IAIA homozygous, then we have IAIB heterozygous, as well as we have IB homozygous. And if you look into the ratio, genotypic ratio, we have 1:2:1. Out of 4 boxes, one has homozygous IAIA, one has IB homozygous, and 2 of the boxes have IAIB heterozygous. Now we need to know F1 phenotype. So we can predict phenotype by our knowledge of the genotype. So IAIA would be A blood type. Both are homozygous. IAIB, we know it is codominant, so it is going to be AB blood type. And IB homozygous, so they are going to be B blood type. So we have in their 4 children, they can be having 3 different types of phenotypes, which are A, AB and B, and this can be in the ratio. What about the phenotypic ratio? Phenotypic ratio is again going to be 1:2:1. So 1/4 of the their children are going to be having A blood type, 1/4 of their children are going to be having B blood type, and half of their children are going to be having AB blood type. So this is how we know about the percentage or the frequency of blood type in the children. Okay, so this is our result or the answer or the solution to this question. So out of their 4 children, 1/4 or 25% will be A, 50% will be AB, and 25% will be B blood type. Okay, so this is how we work out with some of the problems related with blood type. And you can be expecting even such problems in your exams as well. Okay, so I will give you with some of the questions relating with such blood type problems, and you're going to be solving it on your own by using the correct genotype, by using the Punnett Square, and then predicting with the possible genotypes and phenotypes of the children. Now, here are 2 other questions which you can be solving on your own. The first one says, a man who has type B blood and the genotype is homozygous BB is married to a woman with type O blood. The question asks, what blood type will their children have? I guess this is quite workable and easy. The third question is, a person with type A blood, and in the bracket it's given unknown genotype. Okay, this person with type A blood with unknown genotype marries a person with type O blood. The question is asking, what blood types are possible among their children? So I'll just give you a clue here in this question. It's quite easy, but the thing is, when you see that the type A blood of this person has an unknown genotype, we have to work out with both possible genotypes. So we know that type A blood has two possible genotypes, homozygous A genotype as well as heterozygous A genotype. So what you're going to be doing is you're going to be working out both of the possibility separately. Okay, so if this is the, in the first possibility, if this is the homozygous, let us say dominant A genotype, then these are going to be their children. If this is the possibility, the second possibility is heterozygous type A type of the genotype, then their children are going to be having such and such genotype and phenotype. Okay, so that is how you're going to be using both of the possibility or both of the genotypes for this person with type A blood and crossing with the person with type O blood. Yet another non-Mendelian inheritance that I'm going to be taking up is Pliotropy. Now this refers to the expression of multiple traits by a single gene, or in another way we can say that this is the effect of one gene on many traits or several traits. Okay, this is just hypothetical. It need not be always one gene affecting 3 traits. It can be one gene affecting multiple number of traits. So one of the most widely cited examples for Pliotropy in humans is a condition that is called as, or a genetic disease that is known as Phenylketonuria. So I'm going to be taking an example of the disease, which is Phenylketonuria. Phenylketonuria, which is also abbreviated as PKU. Now this is a genetic disease. It's actually a autosomal recessive genetic disease, which you're going to be learning later. So this genetic disease is caused by the mutation in a single gene. Okay, due to a single gene mutation. So phenylketonuria is caused due to single gene or one gene mutation. So it is a defect of one gene, or it is going to be the effect of one gene mutation. So one gene mutation results in, you can say the effect here is, there is lack of or deficiency. This results in the deficiency of an enzyme which is very crucial for the metabolism of phenylalanine, which is known as phenylalanine hydroxylase. So the name of the enzyme is phenylalanine hydroxylase, or it can be also abbreviated as PAH. Okay, so due to a single gene mutation, it results in the deficiency or the lack of, the shortage of an enzyme in these individuals, and the name of this enzyme is phenylalanine hydroxylase. Now we need to know why this enzyme is important for a normal individual. The normal function of this enzyme is, when this enzyme is in enough concentration, they can be helping in the conversion of, so this, it helps in the conversion. Okay, so this is going to be helpful in terms of metabolizing an amino acid which is known as phenylalanine. So this amino acid known as phenylalanine will be converted in the presence of the enzyme phenylalanine hydroxylase into another amino acid called as tyrosine. So for the conversion of phenylalanine into tyrosine in normal individuals, we have the enzyme which is phenylalanine hydroxylase. So this is the normal function of the enzyme. Now, individuals with this genetic disease will not be able to be converting phenylalanine efficiently into tyrosine because of the deficiency or the lack of the enzyme or the non-functional nature of the enzyme. And as a result of which, now such kind of PKU, in PKU we don't have this conversion taking place. There's a block in the conversion of phenylalanine into tyrosine, and as a result of which, these individuals are going to be having excessive accumulation of phenylalanine in their body. And these could be accumulating in the blood, it can be accumulating in the saliva, or it can be also accumulating in the urine. Okay, and as a result of which, because of excessive accumulation of phenylalanine in the body, and because of the inability to be metabolizing phenylalanine into tyrosine, they have many different effects. Okay, and the effects are going to be seen in different organ systems or different of the functioning of the body. Now, some of the major effects are that I'm going to be referring in terms of phenylketonuria will be, now because of excessive accumulation of phenylalanine in the brain, okay, it will be resulting in the retardation in the growth or the development of brain, and this would cause an effect which is called as mental retardation. So you can be observing that individuals with PKU are showing some mental retardation, and that is because of the excessive accumulation of this toxic molecule which is phenylalanine in their brain. Likewise, excessive accumulation of phenylalanine can also be causing the growth or the development of the bone to be retarded, and as a result of which they can also be showing another unique effect which is called as microcephaly. So these individuals with excessive of phenylalanine in their body can also be having unusually small sized head. So the growth and development of the bone and the skull structure is also deformed. Okay, so they have very small sized head which we call as microcephaly. And at the same time, excessive accumulation of tyrosine can also be resulting in, because it doesn't have the synthesis of conversion of the phenylalanine into tyrosine, which is very much important for the synthesis of melanin pigment. So this also can be causing light pigmentation, especially of the skin, light skin or hair pigmentation. So these are some of the common effects of PKU on an individual. And because the defect is only in one gene, but you can be having the effect, or you can be having the kind of the expression of the effect in many of the different organ systems. Such kind of the effect is known as Pliotropy. And this can be inherited in an autosomal recessive way. So it can be if an individual receives both of the recessive defective allele. Okay, if both of the recessive defective allele are received by a individual from both of the parents, then this would be resulting in the expression of the disease which is called as phenylketonuria or PKU. Here is another example of Pliotropy from your textbook. Now this refers to the starch synthesis gene in pea plants. So there's a gene in pea plant which regulates with starch synthesis. And this gene has two alleles, capital B and small b allele. Now pea plants with homozygous capital B type of genotype will always be producing large sized starch grain, and they are also going to be, on maturity, they are also going to be producing seeds that are all going to be having round shape. Okay, so that is how we have the homozygous capital B combination of the alleles, which results in large sized starch grain as well as round seeded seeds in the pea plants. While pea plants with homozygous small b recessive allele, okay, would be resulting in the formation of smaller sized starch grain. And on maturity, these plants are going to be producing wrinkled seeds. Okay, so we have smaller sized starch grain and wrinkled seeds produced by homozygous small b recessive type of pea plants. Whereas pea plants with heterozygous combination of capital B and small b would always be producing medium-sized starch grain and their seeds would be always round in shape. So what we see here is, basing on the nature of the combination of the alleles, we can be having the gene having two effects on the pea plant. Okay, the two effects are, it can be controlling with, or it can be causing an effect on the size of starch grain, as well as it can be causing an effect on the shape of seeds. So size of starch grain can be large sized, medium sized, or small sized, depending on the nature of genotypic combination, and the shape of the seeds can be round or wrinkled. So in this way, we also get to see that one gene is affecting, in this case, two of the different traits. The last non-Mendelian inheritance that I'm going to be taking up is polygenic inheritance. This means to say the additive effect of multiple genes on a single trait. Or in another way, here is one character or one phenotype controlled by two or more than two genes. Now, all of these several numbers of the genes that control a specific trait are collectively known as polygenes. Now, in a polygenic inheritance, the gene which contributes to a trait have an equal effect on the phenotype, and because of which the alleles of each of the gene contributing to the trait can be causing an additive effect, or can be having a quantitative effect on the phenotype of an individual. And with the increase in the number of genes controlling a trait, there would be an increase in the possible combinations of phenotype in the population. Okay, so polygenic inheritance cannot be expressed in simple Mendelian pattern of inheritance. A trait cannot be expressed in two contrasting forms. Okay, so here we have the trait expressed in a range of phenotype, which is called as a continuous variation. We can be finding a continuous variation in the trait in a population because this trait is not controlled by a single gene, it's controlled by two or more than two genes. Let me now consider human skin color as an example for polygenic inheritance. This is because human skin color is showing a range of phenotype in a population. And you might also be observing a range of skin coloration in your own family. Now, this is all because skin color is controlled by many genes. Okay, so skin color is normally considered to be controlled by 4 or possibly more of the genes. But in this example, we are going to be considering or assuming skin color to be controlled by 3 different genes. Okay, and the hypothetical genes that we are having are gene A, gene B, and gene C. And each of these 3 genes are going to be existing in 2 allelic forms. A capital dominant allelic form and a small recessive allelic form. And the dominant allele or the capital letter would be denoting for the dark coloration, and the recessive allele or the small letter would be denoting for the light coloration. So dark coloration is dominant over light coloration. Now, basing on the combinations of these 3 genes in an individual, we can be expecting with a specific phenotype or a skin color. Since we are considering 3 different genes to be controlling with skin color, each of the parents having 6 alleles for skin color would be producing 8 different types of gametes. You can work out if you want by using the Punnett Square idea. So each of the parents can be producing 8 different types of gametes. And with the random combination of these parental gametes, we can be expecting 64 possible genotypes in human population. Now, I'm not going to be working out all of these 64 combinations of the genotypes in the Punnett Square. You can easily see here in the figure. So you have all these 64 combinations, possible combinations of genotype in a human population. Now, what I'm going to be taking up here is of these 64 possible total combinations of genotype, we can be expecting 7 different skin colorations. Okay, from all of these 64 combinations. And these 7 different skin colorations are shown here. In the sense, I'm not having all of the letters denoted for each of the 7 skin coloration. But what I'm having is, I'm having a kind of general depiction of a symbolic depiction of each of the skin color. Now, if you take this skin color, so this is a combination where in this individual or in this particular genotype, all of the genes, all of the alleles of the genes are existing in the dominant state. So all of the letters, since we have 3 genes, each genotype is going to be having 6 alleles. So all of the 6 alleles or letters are going to be in capital form. That's very easy for us to guess. So it's all going to be capital A, capital A, capital B, capital B, capital C combination. So we can be expecting some of the human population to be having such type of genotype. Then we can also be expecting a certain population of human to be having 5 dominant alleles and 1 recessive allele in the genotype. And this 5 dominant allele could be in any combination. Okay, we are not bothered about. If you want, you can be checking into the Punnett Square, the 64 possible combinations in the Punnett Square. But the basic idea here is we have a combination where 5 of the letters of the 6 total letters are capital letters and 1 is small letter. And this is another type of a skin coloration. Next skin coloration is where we have of the 6 total alleles, 4 of the alleles are capital lettered or dominant and 2 of the alleles are recessive or small letter. Okay, so we have 4 capital and 2 small lettered combination of genotype or the skin coloration. As well as we can also be expecting individuals with equal numbers of capital and small letters. 3 dominant letter and 3 recessive letter. Okay, which we can be easily predicting. It is a heterozygous with capital A, capital B, capital C, small c. Okay, then we can also be expecting another form of genotype where we have 4 of the letters are small letters and 2 of the letters are the capital letters. Okay, so this can be even yet another type of skin color combination of genotype. The sixth combination here is we can also be having the genotype with 5 recessive or small letters and 1 capital letter. Okay, and finally we can also be having a genotype which is of all with recessive letters or all with small letters, and this is easy for us to guess. It is going to be small a, small b, small c combination. So in this way, from the 64 possible combinations of genotype, we can be deriving at 7 major skin coloration or 7 major types of genotypes. And these numbers of the dominant allele and the numbers of the recessive allele are going to be determining with the skin pigmentation, the amount of skin pigmentation in an individual or in a population. Okay, so as we said, dominant allele is for dark coloration, recessive allele is for light coloration. So the more dominant alleles an individual is going to be having in his or her genotype, the more darker would be the skin coloration. Okay, so of all these 7 different skin color type, this combination where all of the 3 genes are existing in the dominant state, this combination, homozygous dominant combination, would be having the darkest skin coloration. Okay, so the individuals with darker skin coloration in a population is thought to be having all of the dominant or more of the dominant alleles. So we have darkest, darker, maybe a little darker. Okay, so individuals with such heterozygous combination of alleles will be having an intermediate skin coloration, neither too dark nor too very light. Next, the individuals with 4 recessive alleles and 2 dominant alleles. These individuals are going to be showing more lighter skin coloration because they have more number of the recessive allele to that of dominant allele. Likewise, individuals with 5 recessive allele and only 1 dominant allele are going to be still more lighter because the number of recessive allele is much higher. And finally, individuals with all of the 6 recessive alleles, a, small b, small c combination, they are going to be showing with the fairest or the lightest of the skin color. Okay, so in this way, we get to see here that the gradation of skin coloration or the continuum in the skin coloration depends on the allelic combination. More of the dominant allelic combination would mean more of a darker skin coloration, and more of the recessive combination, allelic combination, would result in a more lighter skin coloration. And individuals with a kind of a heterozygous equal numbers of dominant and recessive alleles are going to be showing with an intermediate or a medium colored skin coloration. So such is the spectrum of skin color that we can be expecting in human population because of the polygenic effect, right? And whenever we try to show the polygenic effect in a population, the distribution of such effect tend to be always showing a kind of inverted bell-shaped curve, where the two extremes having all dominant kind of the allelic combination and all recessive type of allelic combination, or individuals with the darkest and the fairest of the lightest of the skin color are actually in lesser fraction of the population, whereas individuals with the intermediate heterozygous combination, right, capital A small a, capital B small b, and capital C small c combination, they are going to be in the highest fraction of the population. And we can be also having equally more numbers of individuals in the range of dark coloration as well as in the range of light coloration. So that is how we understand with polygenic effect on human skin coloration. Having said that, we need to also understand here that human skin color is not completely controlled by the genetics. We also can be easily understanding with or observing with the effect of environmental factor on human skin color. So the amount of exposure of your skin to the UV light or the sunlight would definitely be having an effect on skin coloration. So the more exposure you have, direct exposure you have to this sunlight, skin coloration would be becoming more darker. That's a kind of the observation that you can be seeing in yourself or in anybody living in more hotter regions. And then people who have lesser exposure to sunlight or people who are living in more colder regions are going to be having a little more lighter skin coloration. So that implies that human skin color is not completely controlled by the genetic makeup of the individual. We also can be having environmental factor having a effect on the skin coloration or the phenotype. Okay, so with this we completed with all of the different types of inheritance which are deviating from, which are non-Mendelian, which are deviating from the Mendelian principles of inheritance. Okay, and you are expected to be noting down each of these differences with specific examples. So with this, I come to the end of today's lesson, and in my next lesson, we are going to be taking up chromosomal pattern of inheritance or chromosomal theory of inheritance.